Good afternoon. First of all, it's a pleasure to be here in this opportunity by Cybernet, where I can share my previews and next works with experts in the neurodegenerative field. So first of all, I would like to thank so much for this organization. And today I will perform this webinar about the protein ADAN10. I will be starting this presentation talk about ADAN10 as a potential blood biomarkers for Alzheimer's disease. This presentation will consist of these steps. So the first one, I will talk about ADAN10, then later results in platelets, results in plasma and CSF, isoforms, about antibodies, next steps, validations in huge databanks, and wrap up. Initially, what is ADAN10? So ADAN10, that is a disintegrin and also a metalloproteinase, is a transmembrane protein. This is a 3D and a simple extra theory of the full ADAN10, so we can observe that it's formed by different and multi-domains, which I would like to highlight the metalloproteinase, that is responsible for its secretase activity. It has so a C-terminal and an N-terminal, and also a prodomain that, when present, inhibits its activity. ADAN10 is a protein of 748 amino acids in length and has several motifs involved in its post-translational regulation process, which are present in almost all the domains of the protein, as shown by these white circles. So, for example, the first one is a PC cleavage site, and also here a cysteine switch, and so on. So many studies nowadays are currently trying to understand these mechanisms with the aim of activating or inhibiting the translation of this protein. In general, ADAN10 it's a secretory route. This is the secretory route of the ADAN10 until the plasma membrane. And in general, it's a metalloproteinase and the main alpha secretase observed in brain. It also can cleavage surface proteins, releasing their ectodomains into extracellular space, so it works as a SHEDAS. It's widely expressed in different cell types, so we can see also in red blood cells, also in white blood cells, in plasma samples, and also in central fluids, for example, the CSF. But ADAN10 is also strictly regulated at the prescriptional, post-prescriptional, translational, and also post-translational levels. Looking for the field of Alzheimer's disease, ADAN10 is highlighted as the most important alpha secretase in neurons. So it acts like a secretase that can cleave the APP protein by the non-amyloidogenic pathway, preventing the beta formation. As we know, the opposite way, the amyloidogenic pathway, is processed by a base 1, that's a base secretase, that also cleaves the APP in specific sites, forming the beta peptides, which can be accumulated and are known as one of the pathological signatures of Alzheimer's disease. So the cleavage of APP by ADAN10 releasing the SAPP alpha, okay, that is a protein fragment with neuroprotective effects. This justifies the potential role of ADAN10 as a biomarker protein, and especially in peripheral media, for example, as blood. And what about the blood biomarkers? Why we're talking about so much about this kind of studies nowadays? So biomarkers have been the major focus of scientific studies in recent years in several areas of research. So in nanomedicine, in aging, in Alzheimer's disease, and also in dementia. And why do you need looking for blood biomarkers for dementia? The blood biomarkers can also assist in the diagnosis. They are less invasive, they're easier, faster, also lower cost, and more accurate diagnosis. They can also predict disease progression, can be detect early changes, for example, looking for preclinical situations. Also evaluate the effectiveness of treatment, so we can use them to monitor, for example, therapeutic response, and also to understand the physiological of the disease. This is a chronobiological biomarkers to Alzheimer's disease, looking for a clinical stage. So as we can see, amyloid plaque biomarkers are dynamic early in the disease, prior to the appearance of the clinical symptoms, and have largely reached a plateau by the time clinical symptoms appears. And we can also see that biomarkers of neuronal injury, dysfunction, and also degeneration are dynamic later in the disease and correlated with the clinical symptom and also the severity of the disease. So as we can see, none of these biomarkers are static. They're very dynamic, and rates of change of each biomarker vary over time and follow a non-linear course, which is hypothesized to be like a sigmoid shaped. So the biomarkers will vary over the course of disease progression. Since 2010, in my master in nursing, I am a research member in the LABEM, is the Laboratory of Biology of Aging in Brazil, which is led by Professor Marcia Comenet, where we have been looking for ADAM10 as a potential blood biomarker for dementia with focus in Alzheimer's disease, especially looking for human platelets and also plasma. About the results we found in platelets, inspired by Italian studies of 2002, I started to investigate the levels of platelet ADAM10 in a Brazilian sample. And we initially observed that its levels were decreased when you're looking for the Alzheimer's disease group compared to cognitively unimpaired subjects. And also this decrease could be observed with the disease progression. I also observed that these levels also positively correlated with neurophysiological tests, as we saw, for example, in the mini-mental examination. It's a cognitive screening tool that we can assess memory, orientation, and other cognitive domains. So the correlation with this test was kind of 0.62. It's a kind of moderate correlation with significant effects. And looking for the association of these variables when we're looking for the mini-mental plus levels of Alzheimer's disease showed highest values in ROC curves. Those decreased levels of ADAM10 was also correlated with clocking drawing test. It's as simple, but it's also an interest test that we can access construction, praxis, physiospatial ability, and also executive function, and also verbal and semantic memory. So I saw that once again, the worst levels, worst scores in the clocking drawing test correlated with lower tests of ADAM10, lower levels, sorry, of ADAM10. And with kind of 0.83 with the correlation and also a significant effect. And the question that followed those results was, what could so be causing this decrease observed in platelets of Alzheimer's disease patients? So we started to investigate it if these decreased levels were caused at a gene expression level. And we have found that no difference in the RNA messenger of ADAM10 between the control when looking for also MCI patients and also within different dementia states and CDR1, 2, and CDR3, which are given by this clinical dementia rating inventory. So those results, which could be seen at first moment as negative, actually just indicated us that the ADAM10 levels was altered somewhere else along its production. So trying to investigate other possible regulations involved in ADAM10 levels, I started researching the expression of microRNAs in whole bloods of people with Alzheimer's disease, compared with cognitive and impaired subjects. And in these studies published in the Journal of Alzheimer's Disease, we performed a huge screening, look for about 700 microRNAs expression. And this study, we found these three most specific ones, the 144, the 374, and 221, that were downregulated in the group of Alzheimer's disease and present significant results. Later, these results were applied in mimics and also inhibitors assays using SH culture cells in order to verify the regulation of these microRNAs in levels of ADAM10. We verified in these cultural assays that the mimic microRNA, specifically the 221, produced a significant downregulation of ADAM10 levels, as the presence of its inhibitory resulted in a significant downregulation, in a significant ADAM10 increased protein levels. So when you block in this 221, we observed a significant increased protein levels of ADAM10, showing us the importance of this specific microRNA for the regulation of this protein. So just as we saw that different conditions and proteins can also regulate ADAM10, we found that its level also are reduced as the disease progression, and that its levels are also increased in people who use serotoninergic antidepressants, for example, compared to people with AD, Alzheimer's disease, who do not use this medication. So we have an increase in patients that are being using these serotoninergic antidepressants. So these results alert us about the influence of also medications on ADAM10, which should be careful considering in that analysis. And what about our results in plasma and also in CSF? Marina Grigoli studied these specific studies looking for and saw that ADAM10 were increasingly in elderly individuals with type 2 diabetes compared to the control group. This level were even higher in the plasma of participants with the presence of the both pathology. We can also observe that several insulin pathway proteins are also changed and also significant with concomitant Alzheimer's disease and diabetes type 2. So these studies indicate that changes in the insulin pathway may be responsible for the increase in plasma ADAM10 levels, and especially individuals with Alzheimer's disease and type 2 diabetes concomitantly, showing that type 2 diabetes is an important risk factor for the development of Alzheimer's disease. In 2020, we also began comparative studies in CSF source in patients with mild cognitive impairment, especially the MCA that are amnestic type. And we observed that in both plasma and also in CSF, EDN10 is increased with the advance of the cognitive decline. We didn't observe a specific difference between MCI and Alzheimer's disease, but for the two conditions, they were different comparative with the cognitive unimpaired. Later, these findings were applied to electrochemical platform for validation, where expressive sensitivity and also specificity values were found through a disposable biosensor. So we developed these biosensors that could detect with huge sensitivity and also high specificity values of EDN10 looking for plasma sources. So we can also observe this increased when we look in different degrees of the Alzheimer's disease and also for different conditions. And this device that we invented had great impact and was also patented in Brazil as an important and possible potential early diagnostic method for Alzheimer's disease. And we're so proud of our work. We were on the cover of this edition. Our studies continue to be published about EDN10 in plasma using biosensors, for example, as using gold nanoparticles, also in different samples with low or with high education levels, also in longitudinal studies, and also, for example, with co-pathologies. Based on our previous studies and others in the literature, we observed some conflicting results, some of which sometimes showed increased levels of EDN10 in platelets. So here, which is an opposite of what we observed. And after some thinking, our group thought that this could be due to different antibodies, for example, epitope binding of the antibodies to different regions of EDN10. So looking for all the regions of EDN10, and also could be about different sources. For example, when you look at different sources as plasma, as platelets, liquor, and for cells. These antibody issues led us to write a great review that were published last year and highlights the importance of the correct antibody usage that I will talk later in this presentation. So one possible explanation about these conflicting results drives on the different isoforms of EDN10. In this moment, I'd like to introduce you to EDN10 isoforms, which are different forms that this protein can acquire during its maturation process. So first of all, we have the immature and the inactive EDN10. It's the pro-EDN10, which has a molecular weight of kind of 85 kilodaltons. The pro-EDN10 down the secretory route has its pro-domain removed and then becomes the mature and activity EDN10. That's about 65 kilodaltons, which then is inserted into the plasma membrane. Lastly, as a regulatory mechanism, we think that the EDN10, the mature form, can be also removed from the membrane by, for example, EDN9 and also EDN15 in a process called ectodomain shedding, which creates this soluble isoform that is released into the extracellular medium, for example, so with kind of 50, 50, 55 kilodaltons. So this is the three isoforms that I would like to introduce you. So this one is the pro-adentem inside the secretory root. When it's become inserted in the membrane, it's kind of the matrix form. And when you have this cleavage with kind of the soluble adentem, so the small portion of adentem, kind of 55 kilodaltons. Okay, and with this nodulate, we tested different antibodies in different biological samples using CSF, plasma, and platelets, and the results show that the CSF and also the plasma samples, a band size of around 15 that could only be detected with the antibody that binded to the extracellular domain. And on the other hand, we observed also a band of 60 kilodaltons of adentem, also present, only present in platelets, which we had seen before and could only be detected with an C-terminal antibody. So this is specific isoform. And what does any of that mean for our studies? With this isoform differentiation, we observed that in the CSF and in plasma, when using the antibody that binds to the extracellular portion, we observed increased levels of the soluble adentem in persons with Alzheimer's disease, which led us to think that maybe in Alzheimer's disease, there is an increase of this ectodomain shedding of adentem in the blood, which explain, for example, the decreased level in platelets and increased levels in the plasma and in the CSF. Lastly, in the same paper, we also tested the enzymatic activity of adentem in these different samples, and we observed very similar results, both in plasma and in CSF. So when you're looking for control group, MCI, and also Alzheimer's disease, the plasmatic and also the CSF adentem, that's soluble form, has little activity when compared to samples that have membrane-bound adentem, as observed, for example, when you're looking for SH cells, when you're looking for platelet samples, and these are a recombinant protein adentem. So this justifies the importance of carefully choose the antibodies for the analysis of studying adentem. As we analyzed in several studies, different antibodies in this review were analyzed, and different antibodies can detect different regions of the protein. So a lot of care and a lot of attention in this analysis are extremely important for carrying out comparative studies, as they can detect, for example, different isoforms of the same protein. The next steps of these studies, the master student, Vanessa, has been dedicated to answer the question, is soluble adentem also inactivity when you're looking for neuronal cells medium, as we saw in plasma and CSF? So with the intention of separating the isoforms to test their activity, we've kind of looking for these preliminary results that the soluble adentem is higher present when you're looking for a cell median, so using a specific antibody for any terminal, and the pro and also the mature form are higher present when you're looking for the cytoplasmic and also for membranes, using the specific also antibody for the C-terminal of the protein. So for this situation, we're using a C-terminal, and for more specific, looking for cytoplasmic and membrane situations. And when looking this, when you use the antibody using for this N-terminal, we can see more expressed for cell median. And this study, we also observed a very feeble activity of A710, the soluble form. So when we trying to compare, for example, to membrane-bound adenine and also the recombinant one, this one is for the membrane, this one for the cytoplasmatic and recombinant. So we can confirm that these results are in accordance with our previous results in plasma and for also in CSF, when you didn't find any activity looking for the adenine soluble form. So in summary, we can hypothesize that adenine in platelets, in SH cells, in neurons, appears to be in its activity form when present in the plasma membrane, and that after cleavage process, among them be adenine or aden 15, for example, its forms becomes inactivity and present in media such as plasma, serum, and also the CSF. I'm currently also developing a second doctorate. It's a collaborative study from Brazil and the University of Barcelona in the Department of Pharmacologic, Toxicologic, and Therapeutic Chemistry in the Faculty of Pharmacy. And the main objective is to associate the protein adenine, but also other proteins from people with Alzheimer's disease, and also looking for other types of dementias. So frontotemporal, also for Parkinson and Lewy-body dementia, in order to build a diagnostic protein panel that could be capable of differentiating Alzheimer's disease from other forms of dementia. The analysis of several proteins in plasma, such as ADAM10, ADAM9, A-beta 42, A-beta 40, tau phosphorylated, and also tau total, and among others that I'm now studying, have already been analyzed among different types of dementia using this ELISA kit. And now I am starting to analyze the association of these proteins. So first of all, I will be performing elastic kinetic analysis, which will be capable to recognize the best options for the protein diagnosis panel in different dementia types. And later, I pretend to apply these results in raw curves combination to perform the sensitivity and also the specificity parameters. I'm also working in a project in the same department at the University of Barcelona with Marie Curie funding. And this project is called Mirada, where I'm looking for blood microRNAs that are adenine targets, looking for novel diagnosis and prognosis biomarkers for Alzheimer's disease, and looking for also apply these biomarkers in electrochemical sensors. So in this project, through bioinformatic analysis, I will search for microRNAs that are targets for adenine and that are also related with Alzheimer's disease, but also with type 2 diabetes. The samples of these studies will be obtained from FACE in Barcelona, and after validation of this specific microRNAs by RT-PCR using Tachman assays, these microRNAs will be applied in biosensors through the research group of the Professor Marie Isabel Curgo in the Autonomous University of Barcelona. So these are initial analysis of the edentem protein in the MIR-target software that I'm performing, which present we can see that edentem has numerous microRNA that can act in its regulation. But when we look at specific microRNAs that related to this both disease, Alzheimer's disease, and also diabetes type 2, we find an overlap of numerous microRNAs as well. However, using this specific software, when I performed a refined selection with the inclusion of the protein edentem in the analysis, and looking for Alzheimer's disease and also type 2 diabetes, according to this software, okay, we found the specific microRNAs, which will also guide the next steps of my study. This is just another figure of this intersection. So we can see using these three combinations. And given what has been present, we have to think why I'm looking for microRNA panels as biomarkers. So microRNA panels, they are greater, they have greater sensitivity and also specificity, so we can use them to combine multiple biological indicators. And as Alzheimer's disease is a huge disease with multiple informations about a lot of biological and pathobiological, we can use them also for early detection. So different biomarkers may become abnormal in different stages of the disease. So they're also important to look for panels of biomarkers. They are so important just to complementary information. So when you're looking and we kind of see a holistic view of the situation of the pathology. They are also important for reduction the false positives and also negatives. So we are trying to increase the reliability of the diagnosis and the accuracy of this diagnosis. The panel is also important for monitoring the progression, so how different biomarkers change over time, as we saw in the beginning of this presentation. And also personalization of treatments. So treatments can be better tailored to the specific needs of each kind of patients. And finally, the accessibility and conveniences, as looking for this kind of biomarkers are less complex and cheaper than when you're looking for imaging methods, for example. Therefore, the current literature indicates us the need for looking for panels studies and panels biomarkers. Building on these previous small sample studies that we perform in Brazil, now we are trying to expand this analysis in larger population. So we are trying to validation this protein as a blood biomarker, but in large population. So the validation of edentem in plasma is being carried out in the city of São Carlos, Brazil, and where the University of UFSCar is located. And through the current project led by Professor Marcia Comenet, this database aims to be formed kind of 500 participants, so from their careers and also older adults. In addition, we are trying to assess several neuropsychological and also biological informations, and also thinking about the future application of these results in biosensors for the diagnosis of Alzheimer's disease. So wrapping up, I would like to finish this presentation saying that ADN10 has decreased level in platelets, increased levels in plasma and CSF. And we saw this analysis in MCI and also in Alzheimer's disease compared to cognitive unimpaired patients. The different forms and the need for specific detection antibodies in future studies. Also important for the validation of these assays in larger data banks. And as we can see, even if ADN10 could not be validated, it's important in the protection against the amyloid plaque for the formation of this amyloid plaque, justify additional studies about this protein. So now we are currently trying to understand another factors that could alter the ADN10 levels and expression. So trying to understand, for example, studies using LDL or HDL levels, increased glucose levels, or microRNAs that I'm performing here, ADN9, ADN15, also in tretazepam and other ones that can regulate the ADN10. So I just would like to greatly thank you, all the collaborators, the members of the OFSCAR and also the UB laboratories, our funding agencies, the Brazilian and also the European ones, and especially the participants and families involved in all these projects. And thank you so much for your attention, and I remain available to share ideas and future conversations. Thank you so much.
All right. Thank you very much, Patricia, for a very comprehensive review of ADN10 biology and also its potential use as a biomarker. So the paper is now open for discussion. As I said at the beginning, if anyone is interested in posting questions, please use the feature of raise hand on your screen, and I will give you the floor to make the question yourself. If by any chance your microphone isn't working, you can also send a question in written using the chat, and I will either read it to Patricia or she will read it herself. It's possible. So while we wait for the first question, let me just remind everyone that CyberNet webinar series are held every other week, usually on Thursdays at 3:30 p.m. And if you're interested in participating as a speaker, please send us a message to Teresa Iglesias, who is the coordinator of the CyberNet training program, or to myself, Miguel Medina, and we will do our best to accommodate a proper slot for you to participate. You can also check the schedule for the next few months at the CyberNet website, and just wanted to remind that the schedule for the second semester of this year is open for suggestion of speakers. Okay, so any questions? I don't see any raised hand. Let me check the chat. Okay, no question coming through the chat either. All right, some questions. Anyone? Don't be shy. Okay, we have raised hand from Maria Salud Garcia. Garcia Young. Okay, please go ahead.
Hi, Patricia. Good presentation. I wonder if you check in cerebrospinal fluid the pattern of immunoreactive bands with another antibody. I have used another antibodies, and my results indicate that we have another immunoreactive band. Did you use another antibody with these samples?
You're talking about the CSF samples, right?
Yes, CSF.
And you looked for ADAM10?
ADAM10, yes, with a Viva antibody.
I didn't understand your...
With the antibody with ectodomain, with antibody that recognize the ectodomain region.
Okay. And you found another?
Another bands, not only in 55.
Which band do you find? Sorry.
I found in 70 chirodaltons, more or less.
70, kind of, it could be maybe the ProADAM10, no?
Yes.
Just looking for the liquid one.
Yes, in cerebrospinal fluid, yes. Did you see the ProADAM10 in CSF?
In fact, for CSF, we just looking for this, and we just see this form, the soluble form of 5,5. I can check it for you if you have my email. We can talk about this. But in this paper I present later, we just find this soluble form, the 5,5 for ADAM10. You're talking, right?
Okay.
Yeah.
Thank you.
All right, thank you. We have a question coming through the chat, and I can read to you, Patricia. It's from Janet Rodriguez. It says, have you studied other ADAMs with high homology to ADAM10, such as ADAM17, among others?
In fact, I'm studying now in this my secondary PhD. I'm studying, and we're looking for also ADAM9. In ADAM17, no, but ADAM9, we're looking for this another protein, but homology protein, no. In fact, no. I think should be very interesting, and because when I performed, for example, assays looking for activity assays, we also block specifically the ADAM10. So we can see also the expression of another alpha secretases, as we are mentioned, for example, this 17. But in fact, we didn't started yet looking for this homology of ADAM10. Now we are just looking for this other secretase that can cleave and inhibits the ADAM10 activity, for example, ADAM15, ADAM90, for example. But it's a good option for future projects. So.
All right, thank you. Yeah, Janet says thank you as well to the chat. Any other questions? No hands being raised. No questions coming through the chat either. So let's make a last call for anyone interested in posting a question.
I remain available for any moment, so we can just talk in email and share ideas, and
all right,
no problem.
That's nice. All right, so things I don't see any hand being raised or any question coming through the chat. I think it's time to close the session here. But not before thanking Patricia once again, her willingness to share her work with us. Thank you everyone for attending, and I wish you really good rest of the day and the week. Okay, thank you everyone.